everyone. In this video, we're going to continue our description of the small intestine by discussing the function of nutrient absorption. So we mentioned previously that the duodenum is responsible for gathering all the secretions from things like the liver, the gallbladder, the pancreas, and of course the chyme from the stomach. But once we move past the duodenum into the jejunum, we're able to talk about the most significant role of the small intestine, which is nutrient absorption. So I would say about 90% of nutrient absorption occurs in the small intestine. And so let's just take a look at the small intestine sort of as a generic structure and just revisit a couple of important concepts. So obviously the small intestine is tightly coiled inside of the abdominal cavity. And the blood flow that arrives at the small intestine is from the superior and inferior mesenteric arteries. And the arteries obviously branch into capillaries, and the capillaries will collect into veins that will ultimately dump into the hepatic portal vein, which delivers the blood directly to the liver. Of course, the parts of the small intestine are the duodenum, which is only about 10 inches in length, the jejunum, which is next, which is about 8 feet in length, and the ileum, which is last, which is about 12 feet in length. So overall, we're looking at just over 20 feet of small intestine, and the intestine diameter is about 1 to 1.5 inches. So it's pretty small, but certainly big enough for the functions. So again, I want to just revisit the layers of the digestive tract, specifically the mucosa, submucosa, muscularis, externa, and serosa. And this time, not to say how it's different, but just to sort of offer a reminder in terms of the layout. So we have these folds called plica, or plicae being the plural version of plica. And these folds actually include the mucosa and the submucosa, and they allow for increasing the surface area of the small intestine, specifically for absorption. And then each, along the mucosae, there are villi, which are small finger-like projections. And so if we just said one, we would call it a villus. And so actually what we want to do really quick is just take one example of a villus and blow it up and sort of take a look at the structure so that we can have a better understanding of what's going on at this level. So in this drawing, we can see some cells that actually make up the mucosal epithelium that are have microvilli on their apical surface. So the apical surface is the exposed surface towards the lumen, and it's abundant microvilli, and then the basal surface is connected to the basement membrane, and then of course we have the lamina propria that's immediately beneath it. And then the other thing that we want to sort of notice into the projection of the villus is the presence of something called a lacteal, and it's actually surrounded by a capillary. So we see the capillary in red, the lacteal in green, and the lacteal is actually a projection of the lymphatic vessel. So we really haven't talked much about lymphatic vessels since we haven't done the immune system yet, but one thing that's really important to the absorption of lipids in the small intestine is the presence of lacteals. So think of it for now as just an extra vessel that is able to collect fluid and absorb other substances, and we'll just sort of highlight that in a moment. So what we really want to do is talk about the differences in nutrient absorption regarding carbohydrates, proteins, and lipids. And we'll notice some similarities between the carbohydrates and proteins and many differences for the lipid absorption. So we'll start by drawing a mucosal epithelial cell. So same thing as we just drew. We have our microvilli on the apical surface, and then we have our lumen that sort of sits at the apical surface. We have our lumen. And one thing we really haven't mentioned before is that the apical surface is actually called the brush border. So because it almost looks like bristles of a brush, we could think of it as the brush border. And then, of course, opposite of the apical, we have the basolateral surface. And then deeper, we have our capillary that's able to absorb some nutrients as they become available. So at the surface, at the apical surface, we have what's called brush border enzymes. So these are enzymes that are responsible for breaking down carbohydrates. Now, we know that enzymes released in the pancreas are able to break down carbohydrates somewhat. So by the time we get to the brush border, what we're really looking at is breaking down the carbohydrates from disaccharides into monosaccharides. So of course it's possible that we still have saccharides that are more, there are more of them than just two sort of connected. But most commonly, if we're looking at something like sucrose, we can't take in sucrose across the apical membrane. So what we really want to do is actually use the brush border enzymes to break the carbohydrates down into glucose, galactose, or fructose, and those are the simple sugars. So we actually can transport across the membrane glucose or galactose as co-transport with sodium via the SGLT1, so sodium glucose transporter 1. So it's like a co-transport passive diffusion mechanism that brings the glucose or galactose into the cell. And then if we're talking about fructose instead, or brush border enzymes breaking something down into fructose, we actually use a GLUT5 transporter. So two different choices for passive diffusion of the glucose or the galactose or the fructose into the cell. And then once the glucose, galactose, and fructose are inside the cell, they actually use a GLUT2 transporter. And the GLUT2 transporter allows for the movement of these monosaccharides across the basolateral surface into the ECF, and ultimately will collect in the capillaries that will deliver the nutrients to the hepatic portal vessel. And obviously there's more to say about what happens in terms of nutrient utilization, but we'll talk about that in a separate video. So let's talk a little bit about the proteins, and we'll notice some similarities here. We have our mucosal epithelial cell with our brush border enzymes at the apical surface. And then we have a couple of different transporters here as well. So the proteins are broken down pretty significantly by the enzymes that are released from the pancreas, but there's still some opportunity for further breakdown here at the brush border enzymes. And so what we'll get actually is co-transport of the amino acid chains, and the amino acid chain can only be 3, 2, or 1 amino acids. So it can't be 4. It's too big if it's 4. So 1, 2, or 3 amino acids can move in as co-transport with hydrogen ions. We can also see that we have an antiporter that's present that exchanges the sodium for a hydrogen. So we're not really making the cell more acidic. We're just sort of moving the hydrogen sort of in exchange. And then when the amino acids are inside the cell, if they are in a chain of 2 or 3, so they have you have a dipeptide or a tripeptide, they're actually broken down by the inside of the cell into single amino acids. We can actually use another transporter to move those amino acids into the ECF, where it will then obviously be collected into capillaries where it travels to the hepatic portal vein. So again, similar carbohydrates and proteins, and then what we'll notice is that lipids are dramatically different. So again, we'll draw our mucosal epithelial cell, and we have our apical surface and our basolateral surface. And we mentioned before that the bile that's secreted from either the liver or the gallbladder, so synthesized in the liver, stored in the gallbladder, secreted via the common bile duct into the duodenum. We get bile released, and then we get a process called emulsification. And so basically what we're doing is taking big lipid droplets and breaking them into smaller lipid droplets so that we can increase the surface area for digestion and make it easier for the different enzymes that are like the lipases to actually break down the lipids. So before we're able to take up the lipids into a mucosal epithelial cell, we actually form something called a micelle. It's similar to if we were to take a bunch of phospholipids and orient them in a sort of sphere so that the hydrophilic portion is facing outward all along the sphere, and then the hydrophobic portion is facing inward. And basically, since we know that lipids by themselves are not able to diffuse easily in a water-soluble environment, the formation of the micelle actually allows for, because the hydrophilic portion is exposed, allows for easy transport of the lipids to the apical surface of the mucosal epithelial cell for absorption. So it's just kind of like a little boat that brings the lipids over to their docking station. But once the lipids are there, the micelle actually dumps them out. And so the lipids move through the apical surface via simple diffusion. And then from there, they'll move to the smooth endoplasmic reticulum for processing. So they'll undergo some modification in the smooth endoplasmic reticulum. And they're also going to synthesize some proteins in the rough endoplasmic reticulum. And ultimately will form something called a chylomicron. So a chylomicron is basically a combination of lipid and protein. And it's actually like a lipid core and then a protein sort of outer coat. And this protein-lipid combination called the chylomicron is enclosed in a vesicle. And eventually the vesicle will fuse with the basolateral surface of the membrane. So we'll get exocytosis of that chylomicron. And the chylomicron is kind of big. And so it doesn't have the ability to be taken up into a hepatic, into a capillary. So instead, it's going to be taken up into a lacteal. So you could think of a lacteal as something similar to like a sinusoidal capillary, where we just have these significantly large gaps in between the cells that make up the vessel. And so it's very easy for a bigger structure to move into, sort of between the cells, into the vessel. And the lacteal actually will dump into lymphatic vessels that will ultimately travel to the thoracic duct, which is basically very close to the superior vena cava in terms of emptying. And so eventually we'll take the chylomicron and dump it into general circulation. So as you can see, there are some notable differences in terms of the absorption of the small of nutrients of the small intestine. But something to keep in mind is that we're basically utilizing either passive transport, so facilitated diffusion for the carbohydrates and the proteins, and then simple diffusion for the lipids. And in all those cases, we're looking at the smallest organic molecules that are able to be taken up. So just only a couple amino acids, or just monosaccharides, or very small lipids. In each case, we're talking about something very tiny that's able to move across the mucosal epithelium, and then ultimately will travel to the basolateral surface and be released for a couple of different possible fates. So certainly we'll continue our next video by describing what happens next in the large intestine, but also we'll pick up in a later video in nutrition and talk a little bit more about what happens after the nutrients are absorbed and are delivered to the liver via the hepatic portal system. But that actually concludes our description for this video. So I hope you enjoyed it. Thanks for watching.